Good morning, everyone. My name is Lauren Malek, and I'm a Bioinformatics trainee in the Myon Lab at the Icahn School of Medicine at Mount Sinai. This summer, I've been building lncRNAlyzr, a web app for performing gene set enrichment analysis on lncRNAs, and today I'm excited to get to share my work with all of you. The motivation for creating lncRNAlyzr was largely due to a request from one of our collaborators, who identified a set of lncRNAs, but didn't really have a tool to analyze their collective functions. Enrichment analysis is a way to analyze groups of genes, and while there's lots of enrichment tools for coding genes, there are much less for lncRNAs. So we wanted to see how we could use common fund data sets to better enable enrichment analysis for lncRNAs. Since proteins are vital to cell function, it was long assumed that because lncRNAs didn't code for proteins, they weren't as important as coding genes. However, recently we've begun to understand that lncRNAs play important regulatory roles in almost all key biological processes. For example, they've been implicated in regulating gene expression, cell development, and they're key drivers in various cancers and other diseases. Our collaborators from the Bester lab at the Technion in Israel identified lncRNAs that are up and downregulated after radiation of cancer cell lines. We were curious how we could develop an enrichment tool for them to see what are the collective functions of the lncRNAs that they identified. The first step in developing lncRNAlyzr was converting common fund datasets into lncRNA set libraries. And this was made possible with a gene-gene correlation data matrix from ArchES-4. This data matrix was provided to me by Giacomo Marino and Alexander Lachman. Using this file, I was then able to generate lncRNA libraries, and we ingested them into a Neo4j knowledge graph database. To perform the enrichment analysis, we developed an independent new web server accessible via an API. When using the web app, the user inputs the list of lncRNAs, and then these are sent to the enrichment analysis server. The server then returns the results, and the results are visualized as a subgraph of lncRNAs and their enriched terms based on the information in the knowledge graph database. To build the lncRNA libraries themselves, we started with CFDE libraries. So for each library, we needed to find which lncRNAs most closely correlate to each gene set. For this, we used a correlation matrix of lncRNAs and the coding genes in each gene set. We'd then take an average of the correlations across each row, which would tell us how much each lncRNA correlates to each gene set. Then, for example, if a gene set had 15 genes, we took the top 15 most correlated lncRNAs, and that became the new lncRNA set. Then we would just repeat this process for all the gene sets in each library. We then serialized the libraries so they could be ingested into the knowledge graph database. Specifically, all of the lncRNAs and the annotation terms became nodes, while the edges connect these nodes if a lncRNA is a member of a particular lncRNA set. So in this example, you can see how every lncRNA set can become a subgraph. The lncRNA set label serves as the source node, and then each of the lncRNAs in the set is connected to it, and the edges are labeled by the library name, which in this case is glygen. As mentioned before, lncRNAlyzr performs enrichment analysis by sending an input lncRNA set to an independent web server. To achieve this, we adapted code from an existing apiter and created a Flask server and an API for it. We request the server to perform the enrichment analysis by sending the lncRNA set plus the library name we want to use. The server then returns a JSON file with the results. There was also the task of setting up the lncRNAlyzr UI web app. To accomplish this part, we adapted the UI developed for the CFDE gene and reprotox partnerships developed mainly by Errol Evangelista from our lab. This UI was developed with Next.js and React. There are a number of features on the site, for instance, one for enrichment, another for browsing the knowledge graph database. There's also a tutorial page. Lastly, for displaying the subgraphs, we use Cypher to query the Neo4j database and then visualize the subnetwork with Cytoscape. That brings us to the final product, lncRNAlyzr. Here, I entered a set of lncRNAs identified by the Bester lab to be upregulated after irradiating a cancer cell line. When performing enrichment analysis, we can choose up to 5 different libraries. For this example, I selected chia3, Trust, GWAS catalog, MGI mammalian phenotypes, and Reactome. Once we submit the set for enrichment analysis, lncRNAlyzr returns a subgraph with the different terms from each library displayed in their own color. You can also view the enrichment results in a table or a bar graph. Looking at the bar graph here, we observe that many of the enrichment terms listed are associated with erythrocytes and erythropoiesis. For instance, from the chia3 library, we can see that tau1, a transcription factor known to regulate erythrocyte maturation, is the top enriched term. We can also see the terms lymphoma and anemia and other terms related to red blood cells. In sum, our results suggest that this lncRNA set is enriched in genes related to erythropoiesis and regulated by tau1, thus illustrating one instance where using lncRNAlyzr was useful to gaining insights into lncRNA function. lncRNAlyzr certainly has room for improvement, though. Currently, it supports enrichment analysis against 33 libraries, and this can be expanded. The UI can also be improved by adding a feature that will enable users to upload lncRNA sets as files. Adding coding genes to lncRNAlyzr is another potential feature, since lncRNAs are likely working together with coding genes, so considering them together would be useful. We'd also hope to see the experimental validation of predictions made by lncRNAlyzr. So for instance, the enrichment analysis results of the example lncRNA set from Dr. Bester's lab could be validated by testing the involvement of the tau1 transcription factor in response to radiation. Finally, I'd like to thank the members of the Myon Lab for all of their help this summer, and thank all of you for listening. You're welcome to put any questions you may have in the Q&A. And I'd now like to introduce our next speaker, Adam Lalani, who will be discussing how he developed an app which shares some of the same technologies I used to create lncRNAlyzr to construct subnetworks from input gene sets and background databases of protein-protein interactions. Thank you.